Okay, so how similar and different are mitosis and meiosis? Mitosis and meiosis are similar because both result in an ending state of cytokinesis, in which the cell divides into two. Both contain prophase, metaphase, anaphase, and telophase, where chromatin turn to duplicated chromosomes, or in meiosis 1's case, they form homologous pairs. However, mitosis is used in asexual reproduction, whereas meiosis is used in both sexual and asexual. Meiosis also contains a part 1 and 2, whereas mitosis is 4 to 5 steps rather than 8 to 9, based on whether interphase is included. Mitosis results in identical cells, and meiosis results in genetically different cells. Mitosis results in 2 cells, and meiosis results in 4 cells. Lastly, crossing over only occurs in meiosis. So, which is more like mitosis? Meiosis 1 or meiosis 2? Meiosis 2 is more like mitosis because there are no homologous pairs in either, unlike meiosis 1, as well as no crossing over. In both, duplicated chromosomes line up on the equator of the cell, and spindle fibers separate the sister chromatids. Both meiosis 2 and mitosis are extremely similar. Now, onto our demonstration. First, duplicated chromosomes find their homologous pairs, as shown here. Next, crossing over occurs. This is the stage in which genes from each homologous pair are shared. This is what helps result in genetically different daughter cells at the end of meiosis. Here, in our example, the different shapes on each chromosome represent the different genes that are being shared. Later on, we will show you how this crossing over, along with a few other factors, lead to differently genetic daughter cells. Now, each homologous pair will line up at the equator of the cell, in any order what they would like. As all this is occurring, the spindle fibers attach to the centromere of each duplicated chromosome and begin to pull away. Now separated, the spindle fibers begin to disappear and the nuclear membrane begins to form again. So, what are some possible errors of meiosis that could be described in our model? A potential consequence of meiosis could be the uneven breakup of homologous pairs or duplicated chromosomes during anaphase 1 and 2, shown above. This would result in 2 uneven daughter cells, where one has an extra chromosome and the other is short one. If one of these abnormal cells were to be fertilized, possible birth defects and problems could occur. Now, as telophase occurs, a cleavage furrow forms, showing the 2 different cells. The final part of meiosis 1 is when cytokinesis occurs, finally splitting the cell into 2. Now, time for meiosis part 2. The duplicated chromosomes now floating around in each cell begin to line up at the equator. Now, during anaphase 2, the duplicated chromosomes are split up by sister chromatids and each are separated each side of the cell. This can be shown above. However, the hands representing the spindle fibers separating them will normally pull all at approximately the same time. In meiosis 2, as you can tell, there is no crossing over. Now, during telophase 2 again, the spindle fibers begin to disappear and the nuclear membrane begin to form again. Each cell pinches a little bit to form the cleavage furrow. Now, for the last part of meiosis 2 and meiosis as a whole. Cytokinesis occurs in both cells, forming 4 different daughter cells. Each of these daughter cells, as you can see, are diploids. These cells, many a times, can be considered sex cells. So, finally time to describe what independent assortment is. Independent assortment occurs in which the homologous pairs line up and can be separated in two different directions based on the order they line up in. The number of possible different genetic gametes can be found using the formula 2 to the power of n, where n represents the number of homologous pairs lined up. In our case, 2 to the power of 3 would result in 8 possible different genetic gametes. And there you have it, folks. Meiosis 1 and Meiosis 2.